COVID-19 is a virus which has made all of our lives quite miserable. It reared its ugly head on December 19, and fuck me, who knew it'd be this bad, huh? It's a respiratory virus which enters the body through the mouth or nasal cavity and begins to infect the lower respiratory tract. As the virus travels down our respiratory tract, it enters the lungs and begins to interact with cells to enter them. When it reaches the cells, it interacts with a very common receptor on the cell surface called ACE2. A receptor so common that the news made bank telling you that COVID was crawling up your butt just because it was tested there. The virus uses a specialized protein on the surface to infiltrate the cell, the famous spike protein. The spike protein is made up of two subunits, S1 and S2. Both are specialized in certain ways, allowing them to carry the function of a spike protein. Let's first look at S1. Containing the receptor binding domain and the N-terminal domain, this part is used to attach to the ACE receptor. S2, on the other hand, is used for viral fusion and entry, and is made up of parts called the fusion peptide, the hepatopeptide repeat sequence 1 and 2, a transmembrane domain, and a cytoplasmic domain fusion. Once the receptor binding domain binds to the ACE2 receptor, a body enzyme called Temprase2 cleaves S2. This exposes a section full of hydrophobic amino acids, which quickly bury themselves into the cell membrane and begin the fusion with the cell membrane. Following this, the extended spike folds onto itself and forces the cell membrane and the viral membrane to fuse. Once the viral RNA is pushed inside, it begins carrying out different tasks to transform the cell to the virus's liking, like a terrible houseguest. The virus begins to transform the endoplasmic reticulum into a structure made up of many different bubbles called double membrane vesicles, providing a safe haven for the viral RNA to be replicated and translated. But before this, it has to eliminate the competition. The virus is a protein called NSP1. It recruits proteins which travel around, breaking up RNA strands which don't present the viral tag. It also physically blocks the channels into the ribosome so that no human RNA can enter and create new proteins. Finally, NSP1 also shuts down the alarm system of the cell by stopping mRNA from getting out of the nucleus. This prevents new proteins meant to alert the immune system from getting out of the cell, essentially shutting down the entire cell. When our immune system does find the cell, it begins an overblown immune response leading to a cytokine storm, causing huge amounts of damage. To evade this, the infected cell begins to express the spike protein and activate calcium ion channels, expelling a fatty layer onto the outside of the wall, similar to the layer which sticks muscle cells together. This creates a mass of around 20 cells, all with ACE2 receptors helping the hijacked cell stay clear of the immune system. Once this is done, ribosomes translate two sections of viral DNA, which are cut into 16 proteins, followed by more RNA translation that creates a strand of 26 proteins. Through this, new structures are made which allow viral structures to form into viruses. From here, a human enzyme called furin makes a cut on the spike protein of the virus, priming it for another target. This is the basic attack strategy by COVID. Different variants will have most of the same attachment methods. However, a protein here, a protein there, and suddenly it's much more effective at its job and much more ineffective. It's important to understand a mutation doesn't have to be dangerous. Most mutations help the virus survive longer, which means either more effective, better at evading the immune system, and generally less deadly. Before talking about the Delta variant, it's important to note that this is early data and will inevitably change. It would appear that Delta is twice as transmissible as the original Wuhan strain. Moreover, it would appear that viral load seems to be 1,000 times higher. It would appear that the virus isn't any more dangerous. However, it isn't any less dangerous either. As a result, it has managed to fill up hospitals faster as a result of its ability to spread so suddenly, and it appears to be slightly resistant to certain vaccines as well. The main mutation that affects this variant are in the spike protein. A mutation on the 614th position leads to a change of amino acid from aspartic acid to glycine, exposing the multi-basic furin-like domain and producing a more open configuration in the activated receptor binding domain. There is also some evidence to suggest that it also increases the speed of the reverse transcriptase. It also works with another mutation at the 859th position and forms a promoter hydrogen bond and brings the residue of S1 to the residue of S2, as well as eliminating a hydrogen side chain and increases main chain flexibility by altering promoter interactions. Moreover, it looks like the 614th mutation increases viral membrane fusion. As the mutation is right next to the fusion peptide, near the TMPRSS 2 cleavage site. This destabilizes it and allows easier binding. From the alpha variant, there appears to be 3 different protein mutations, leading to the receptor binding domain having an easier time to attach to the ACE2 receptor. At the antigenic supersite, there are also a number of mutations occurring, which altogether help the virus evade the immune system. There are also a couple of silent mutations of cytosine to thymine at positions 3037 and another at position 14,408, with this one affecting the RNA polymerase. Thank you for watching. Please like, subscribe, and share the video if you learned anything at all. I'm trying to increase the quality of the videos, so stay tuned and watch the channel grow. It'll be more satisfying. Thank you.